So I'd like to introduce Dr. Erica Claud to speak about probiotics and the neonatal microbiome. And Dr. Claud is currently a professor of pediatrics at the University of Chicago, and she completed her medical education at Northwestern and stayed for her residency and fellowship at Lurie Children's Hospital. Her research focuses on intestinal epithelial biology as it relates to neonatal necrotizing enterocolitis, and her work has been funded extensively by March of Dimes and the NIH. She is a dedicated educator and author and has published several articles as well as written chapters in leading textbooks on pediatric gastrointestinal disease. Thank you for being part of the ONTPD Online with the Experts series, Dr. Claud. And just before you start, I want to remind everyone to please stay muted, and we'll have a Q&A session following the presentation. And I'll hand it over to you now.
Okay, thank you so much. Thank you for that introduction. It's a pleasure to be here. I will be speaking about probiotics in general, which is sort of one of my favorite topics, but I think also a bit controversial. So my goal is to put probiotics in a larger context and then also highlight key articles on this topic so that you're familiar with them as well. So we'll be discussing some older articles as well as some newer articles. So when we think about bacteria in the neonatal intensive care unit, I think that it's frequently viewed as a battle between good and evil. So pathogens are evil. So pathogens we understand. This goes back to Koch's postulates. A microbe is suspected of causing a certain disease. If that is the case, it must be found in every case of that disease. The suspected microbe must be isolated and grown in the laboratory outside of the host. And when this suspected pathogen is introduced to a healthy, susceptible host, that host must develop the disease pretty much every time. And then that pathogen must then again be obtained and re-isolated from that experimental host. So we have a lot of specific understanding of pathogens. So that's the evil side, so we say. The good side is commensal microbiota, right? But this is vaguer, not quite so specific. These microbiota as a group supply essential nutrients. They provide colonization resistance. So if you have a properly working commensal microbiota, it's more difficult for pathogens to gain a foothold. And they affect host development, both the intestinal development and the immune system. So if we have this battle between good and evil, we can then think about our tools. So we have a couple of tools. So obviously antibiotics, we're familiar with this. The antibiotics are supposed to take care of the pathogens. And then the counterpart of this is then, so it seems, probiotics. So probiotics are supposed to accentuate or improve the commensal side. Probiotics are live microorganisms that, when administered in adequate amounts, confer a health benefit on the host. So these are tools. These aren't really good or evil, but I think if you're going to use a tool, you need to understand it. So again, we understand antibiotics well. We understand there's concerns. We want to be sure that we take into consideration safety. And what is the optimal antibiotic based on susceptibility and narrow versus broad spectrum? We want to make sure that we have optimal administration, that we understand what patients, dose, timing, length of treatment. We need to understand mechanism of action, standardization of preparations, and potentially whether or not there's a concern for antibiotic resistance or an impact beyond a particular patient. I think frequently we think that probiotics are either neutral or good, and we don't recognize quite as frequently that the same concerns exist, that there's concerns of safety, that we should know what is the optimal probiotic organism for a particular condition. We should understand who should receive this and under what protocols. We should understand the mechanism of action as well as standardization of preparations. But we've kind of, I think, skipped over some of these concerns because the literature has been compelling. So probiotics were initially introduced around necrotizing enterocolitis. And these are some of the historic studies that you should all be familiar with. The first study is actually from 1999. So this is Hoyos et al., a study from Bogota, Colombia, with 1,237 infants. They took all admitted newborns, gave them probiotics, and compared them to historical controls. The probiotic administered was Infloran, which is a combination of Lactobacillus acidophilus and Bifidobacterium infantis, and there was a decrease in the incidence of NEC to 2.9% compared to 6.6%. So a 50% reduction. This was very compelling. But there were concerns. It was all admitted newborns. It was not randomized and controlled. At the same time, it was historical control. So in 2005, Lin et al. Did a study in Taiwan. This time, 367 infants, very low birth weight infants, less than 1,500 grams. This was prospective, blinded, randomized, same probiotic. Again, a decrease in incidence from 1 to 1.1% from 5.3%. That same year, Bin Nun et al. Published from Israel, a different probiotic study with Acinetobacillus, Bifidobacterium infantis, Strep thermophilus, and Bifidobacterium bifidus, decreased to 1% versus 14%. So I think, a key thing to note is we're now comparing probiotic studies, but it's a different probiotic this time. And then in 2008, Lin et al. Did an extension of their previous study, again in Taiwan, this time multi-center, again with Infloran, a decrease in the incidence of NEC. So this was all very compelling. There were a lot of reports that should definitely give probiotics. Perhaps it was unethical to withhold probiotics. And studies have continued. So just so that you are familiar with them, ProPrems is a significant study, this time B. infantis, Strep thermophilus, B. lactis, from 10 perinatal centers in Australia. Of note, all of these babies had high exposure to human milk. That was a little different from the previous study. Again, infants less than 1,500 grams, multi-center, randomized, controlled. Decrease in NEC from 4.4 to 2% overall. Of note, there was no difference for infants less than 28 weeks gestational age and a birth weight less than 1,000 grams. For this trial, there was no difference in sepsis or mortality. This is the only trial that really had sort of long-term follow-up of these patients with comparable rates of survival without major neurodevelopmental impairment at 2 to 5 years old, but this only was able to capture 67% of the original cohort. A couple other probiotics for you to just be familiar with. Avivo is Bifidobacterium longum. This is the one product that is specifically being marketed to preterm infants. Some of you may have received emails about this. They've been pretty aggressive about their marketing. This time, this is just a single strain probiotic. It's not like the others that are combination. They have shown a decreased incidence of NEC, but with a historical control again. They've shown some other benefits, increased weight gain among severely malnourished infants in Bangladesh. But of note, this works best when there are human milk oligosaccharides present. So it's recommended to be given with breast milk, which isn't always the case for our preterm infants. Of note, it is not FDA approved. It is being produced under generally regarded as safe, kind of like a food product. And that continues to be another concern about probiotics, that they are not very well regulated at this point. And then the last trial that I just want to familiarize all of you with is PIPS, or probiotics in very preterm infants. This is from the UK, another single strain probiotic, Bifidobacterium brev. Again, multi-center randomized. This time there was no difference in NEC, sepsis, or death before discharge. And of note, there was cross-contamination in 37% of the infants assigned to placebo. So whereas in antibiotics, we might think about antibiotic resistance, kind of the counterpart to probiotics is that once it's introduced into a nursery, it tends to extend. So you have this organism across the nursery, whether or not it was intentionally given. So there's certainly sort of variations in how people view this literature. It seems that the outcomes are very promising, but again, the majority of these trials have been outside the United States, and have involved a number of different types of organisms and have all been lumped together. And in addition to this, there's certainly an infectious risk. So this is one publication that specifically described a case series of bifidobacterium longum in preterm infants on probiotic therapy. And then this one was quite concerning. There was a fatal gastrointestinal mucormycosis in an infant following the use of contaminated ABC-Dophilus powder. So at present, there is no FDA-approved probiotic product. Even the ones used in the trials are not generally available. But still, I have heard from others that since the data in terms of the outcomes are so compelling, in fact, the data is more striking for probiotics in NEC than pays routinely without a thought, such as surfactant for RDS or cooling for HIE. Why is there the concern? So one of the things I think is a big concern is that we don't have any criteria for who should receive the therapy. There are no biomarkers for NEC or clear indications for who should receive the probiotics. We would never give surfactant to all preterm infants. There's very specific criteria that indicate which babies are at highest risk for a condition and therefore which babies should receive surfactant. Similarly, we would never cool all full-term babies, very specific criteria. But the criteria for probiotics is merely all preterm infants of a certain gestational age or birth weight from birth long-term to discharge or 36 weeks. So for these patients, yes, there's a decreased incidence in NEC, but I think it would be important to consider another number. So this is the number of patients who received the probiotic in each of these trials who perhaps were not at risk of NEC. So if you take 100% and subtract baseline incidence of NEC for patients who did not receive the probiotic, this is the number of patients that are receiving the probiotic. So for these patients, they're just getting the long-term effects. They're just getting the side effects. And so I think a question is, should we give probiotics to a patient who was never going to develop NEC? Or if we turn it around, in pediatrics, is there a time where we give a therapy across the board, one that we think may impact the immune system, may have a long-term effect, in order to prevent disease in a few? So just think about that for a little bit. I always prefer a more interactive in-person session, but just think about that a little bit because we do. And I think that's vaccination. And I think that should be the standard against which we are comparing probiotic use, not necessarily cooling. So this is old, this is from 2013, but it still holds. The safety and effectiveness of vaccines are under constant study because vaccines are designed to be given routinely during well child care visits. They must be extraordinarily safe. Safety testing begins as soon as a new vaccine is contemplated, continues until it is approved by the FDA, and is monitored indefinitely. And this is not the standard to which we are holding probiotics, even though they're being given to many children who may not have the disease that they are supposedly being treated for. And this is something that could have a long-term impact on them. So let's think about this differently. Instead of thinking about probiotics as good in a battle of evil versus good, let's put this in the context of the microbiome as a whole, because I think that's really what we're trying to get to. And as we understand this more, this is really what we're trying to influence. So the microbiome is all the microbes in and on the body. This is bacteria, viruses, and fungi. There's actually equal numbers of microbes and human cells. Some might even say that there are many more microbes than human cells. So this is a significant element of health. Microbes comprise greater cell numbers, as I mentioned, but also significant weight, metabolic potential, genetic content. So in many ways, human health depends on microbiota health. And therefore, it's really important to understand the microbial community perspective. So again, I don't think this is good versus evil. This is a community. Another way to think about it is as an ecosystem. So just in the same way you might think about a forest or a pond, the microbiome healthy community ecosystem would be notable for high diversity of species, balance, functional redundancy, and resistance to disease. Whereas a sick ecosystem would have a low diversity of species, imbalance, functional disability, and susceptibility to disease. And I just want to highlight this word functional, because it's not only who is present or what are the names of the trees or the organisms in your ecosystem, but what are they doing? So I just want to give a simple illustration of community dynamics to maybe illustrate what I'm talking about. So think about 3 organisms. We're just going to call them A, B, and C. They aren't pathogens or commensal. They're just 3 organisms in a community in balance. So let's imagine that A produces something that B needs, and that B and C share a substrate. When A is present to deliver what B needs to grow, B grows, competes with C for this substrate, and C is then kept in check. And this is balance. But imagine something comes along that perturbs this system. So let's look at antibiotics. Let's say that you have an antibiotic that takes out A. So now A is no longer producing this substance that B needs. B disappears or gets smaller and is no longer able to compete with a food substrate with C, and C becomes more dominant. C isn't necessarily a pathogen. It wasn't necessarily going to cause disease, but in a system that is out of balance, if it becomes more dominant, it's in a place where it can cause a harmful effect on the host. Or let's say there's an alteration in diet so that this key substrate goes away. Maybe a baby's made NPO. So if that substrate goes away and B and C are no longer able to thrive, A is still going to be producing its substance. But now B is no longer present to take it up, and A might actually be causing disease because of this substrate or this substance that's able to impact the host. Again, not because A in and of itself is a pathogen, but because the community is out of balance. And you can imagine what might happen to a community if, say, you now introduce a new organism, because one is introduced into the nursery. If you give a probiotic, it's really going to have a differential effect depending on what is the underlying community. So keeping that in mind, that we're really talking about community dynamics, I just want to introduce a couple concepts about how we study the microbiome and try to understand this community. So some of you may be familiar with this already. We begin with a microbial community sample. Frequently, this is a fecal sample, but increasingly there are more and more studies being done on lung samples or skin samples. It's important to understand the community of origin. You extract DNA, and one approach for studying the community is called the 16S base approach. So for this, we just amplify the 16S ribosomal RNA gene. This is specific for bacteria. Humans don't have it, fungi don't have it. It is a way of enabling us to look at only the bacteria. But it's only one gene. It's not amplify the one, compare it to databases, and then we can get an idea of what is the community composition, what organisms are present. Is called the shotgun metagenomic approach. For this, you again extract DNA from the microbial community, but you break up all of the DNA so that you don't know which DNA comes from which individual microbes, but you can study the whole community and you can get an idea of what all of the genes are. These are again compared to various databases. But this now gives you an idea of function. What can the community do? What do all of these genes look like in terms of pathways? We don't necessarily know which genes came from which microbe, but we understand the function of community. And the next step after this, which isn't really on this slide, we can also look at metabolites. So what is the community producing? What are some of the metabolites, kind of like what A was producing, that might have an impact on the host? This again gets at the idea of what is the function, or what are the communities doing. For the preterm infant, I think it's important to understand how the microbiome is beginning to be developed. It's controversial whether or not there is a microbiome in utero. Regardless of how you view that question, certainly the microbiome begins to be established and increases in complexity after birth. So for our preterm infants that are starting to be our patients right at birth, how this develops over time is important. And the microbiome is impacted by the environment. So for a baby, obviously the first big change in environment is mode of delivery. This is a landmark study by Gloria Dominguez Bello from 2010. It looked at mode of delivery and found that babies who were born vaginally had a microbial community that was similar to the maternal vaginal community, and babies who were born by cesarean section had communities that were similar to the skin microbiome of their mothers. Now, this is full-term infants. Another key environmental factor is type of feed. So there's differences between breastfed infants and formula-fed infants, with breastfed infants having more bifidobacterium and formula-fed infants having more organisms that are perhaps concerning, Staphylococcus, E. coli, and Clostridia. And then there's a difference by gestational age, with full-term infants having a wide range of anaerobic and aerobic organisms by 10 days of age, but preterm infants having delayed colonization with fewer species that are concerning for being more virulent. And this is probably not a big surprise because the NICU environment influences the microbiome through things such as whether or not the babies are being enterally fed at all, whether, again, they're receiving breast milk versus formula, all of the instrumentation, the endotracheal tubes, the NG tubes, and then certainly all of the medications. Antibiotics are commonly thought of as influencing the microbiome, but also H2 blockers and opioids and many of the other influences on the baby are all going to impact the microbiome. So rather than look at just probiotics and NEC, let's take a broader view and now look at the microbiome globally and NEC. So I think it's somewhat accepted that one of the risk factors for necrotizing enterocolitis is bacteria. Many of the studies up until now have looked at culture, and there have not been differences between the microbial community of babies with and without NEC by culture. But it was hoped that some of these tools, like 16S ribosomal RNA gene-based analysis, would help us understand NEC better than we have before. So this was an early study from my lab in 2009, 16S ribosomal RNA gene-based analysis of fecal microbiota in preterm infants with and without NEC. We looked at samples from 20 preterm infants, 10 with NEC and 10 matched controls, including 4 twin pairs from a single NICU. And these were all right before NEC diagnosis. So we had been collecting longitudinal samples, and for the babies that went on to develop NEC, we took the sample right before diagnosis and then took a control sample from a similar infant of similar gestational age and day of life. So the first thing we found was that the NEC patients did have lower diversity. So going back to our community dynamics, decreased diversity is associated with an unhealthy community. So disease, unhealthy microbiome community, that fits. And then when we looked more specifically at the organisms present, Proteobacteria is a phylum. Control infants had about 40% Proteobacteria. The remainder, about 60%, was of other phyla, whereas our neck patients had almost the full community as proteobacteria, less than 10% other phyla. And of note, all of these numbers would be considered very abnormal for an adult. An adult is expected to have less than 10% proteobacteria. So there are differences in the overall microbiome community for babies with and without neck. But what this shows is individual patients, top panel is neck, bottom patient is control, and the dominant genus is labeled. And what you can see is there might be a difference in the overall microbiome community, but the organisms aren't the same. This suggests that there isn't a specific pathogen that is causing necrotizing enterocolitis. But each of these neck patients you can see has a bloom of a specific organisms. These are all gamma proteobacteria. We don't see this in the control patients, except for these two. So there were two patients that were put in the control group based on a variety of factors. They did have a bloom of this gamma proteobacteria, Escherichia. And interestingly, they both did go on to develop neck in the next 2 weeks. This is another way of looking at the data. This is a PCA plot. So each point represents a microbial community, a full microbial community, actually in 3-dimensional space. This is obviously a 2-dimensional graph. So the points that are closer together are more similar to each other, and points that are further apart are more dissimilar. And you can see that the blue square control patients cluster together, the red dot neck patients all cluster together, again, except for these two control patients, the cluster with the neck patients. And again, these were the two that went on to develop neck in the next couple of weeks. The one clinical factor that we found to be associated with these patterns was number of days of antibiotics prior to the sample being obtained. So the neck patients had had many more days of antibiotics before we analyzed their sample. So this was all a difference in the microbiome right about the time of disease. That's sort of too late to intervene. So the next thing we looked at is, when does the shift in the microbiome occur? So now we did analyze the longitudinal samples over time. And what we found was that for patients that went on to develop neck, or those who remained healthy, early samples were actually pretty similar to each other. See, these are closer in space. But with the healthy patients going on in one direction and the babies who went on to develop neck following a different trajectory. And at this point, we kind of had two options of what we could do. We could go on and try to figure out what was going on with the babies who developed neck, or we could go on and try to understand what was different about the babies who remained healthy. And that's what we decided to do. We decided to look a little more closely at the babies who remained healthy. So for this study, we looked at samples from all the babies who did not develop neck and compared them to longitudinal samples from a healthy, full-term breastfed infant cared for at home. So everyone, I think, would agree that that's the gold standard of an ideal baby. But you can see that all of these samples cluster pretty closely together over, say, an 8-week time frame, kind of an average time course for a preterm baby to be in the NICU. And this isn't what we saw for even the preterm babies who went on to remain healthy. So for the preterm babies, there was a cluster right around 2 weeks of life, then 3 to 5 weeks of life. And it was only after 6 weeks of life, these open squares, that the samples begin to converge on the full-term breastfed infant. So there seemed to be a developmental pattern to the microbiome. This probably shouldn't be a surprise to us. Everything is developmental in the NICU. But just think about it. If you're introducing a probiotic, that's not changing over time. Certainly the protocols up until now are the same probiotic at the same dose from birth across time. It's not something that allows for this developmental progression. This is another study that looked at samples from 58 patients who, again, were considered relatively healthy, again, by 16S ribosomal RNA sequencing. And what they found also was that there was a known progression of the preterm infant microbiome from bacilli to gamma proteobacteria to Clostridia, and that clinical factors such as antibiotics and breastfeeding altered the pace, but not necessarily the pattern. So this again suggests that there is something that's developmental that is normal and perhaps important. This didn't involve probiotics, and still the microbiome is changing. I think it's interesting that there are specifically antibiotics and feeding type mentioned. These are certainly other factors that have been shown in other studies to impact the microbiome and have been specifically associated with NEC. So again, just to highlight classic studies that hopefully all of you are familiar with, but kind of fit into this topic. This study by Cotton et al. From 2009 looked at prolonged duration of initial empirical antibiotic treatment and rates of NEC and death for extremely low birth weight infants. This was a retrospective cohort analysis of ELBWs from 1998 to 2001. Over 5,600 infants who had been admitted to 19 centers, and they analyzed about 4,000 who had survived more than 5 days and received initial empirical antibiotic therapy, but had sterile culture results in the first 3 days. So babies who perhaps had received empirical antibiotic therapy without needing it. The outcomes of primary interest were NEC or death, NEC alone, and death alone. What they found was there was a median therapy duration of 5 days with a pretty significant range. About half had received prolonged empirical therapy, so more than 5 days without a positive culture, and that this prolonged therapy was associated with increased odds of NEC or death. In fact, each empirical treatment day was associated with increased odds. So it was dose response. There was a dose response. And the conclusion was that prolonged initial empirical antibiotic therapy may be associated with increased risk of NEC or death and should be used with caution. And their thought was that the choice of antibiotic duration is likely to affect colonization of the neonatal intestine. So they didn't look at the microbiome, but their thought was that was what was being impacted. And that colonization, at least in animal models, seems to contribute to the physiologic development of the intestine, immunologic system, and absorbance of nutrients, and alterations and variations in any or all of these areas could increase the risk of NEC. So that's one study. The other kind of classic study then looks at breast milk, again from 2009, the role of human milk in extremely low birth weight infants and risk of NEC or death. This looked at about 1,200 ELBWs and the NICHD Neonatal Network glutamine trial from 1999 to 2001. There were 15 centers. The primary outcome was NEC or death after 14 days. And what they found was that the hazard of NEC or death was decreased by 13% for every 100 cc's per kilo increase in human milk. This is mother's own milk. This was not donor's milk. An infant received in the first 14 days of life. Increasing the amount of human milk within the first 14 days of life was associated with increased survival time free of NEC, and increasing the proportion of human milk to total intake, enteral plus parenteral, was also associated with decreased relative risk of NEC. So I think these results aren't a surprise either, but I think just looking at this, one can think about prolonged antibiotics and breastfeeding in comparison to probiotics. So these are all modifications that happen early in life. So probiotics are started as soon as babies start feeding. The breast milk data was from the first 14 days of life. The prolonged antibiotics was anything more than the first 5 days of life. We're really looking at alterations early on. And you can see that these potential alterations to the microbiome, if we're thinking that what we want to do is modify the microbiome to decrease NEC, could occur by giving probiotics, but it could also occur by decreasing antibiotics or by giving any of mom's own milk in the first 14 days of life. And one could argue that decreasing antibiotics or giving breast milk are both better studied and safer ways of modifying the microbiome than probiotics. And I think it's also important to remember that when we are modifying the microbiome, we are modifying it for the long term. And that as our infants are developing, the microbiome is also developing, and it's not just all about neck. One thing that has been studied quite a bit recently is the microbiome gut-brain axis. This has been shown in adult studies and is beginning to be studied in infants and children as well. There seems to be a connection between the central nervous system and the gut, and potential mechanisms by which this occurs are through immune modulation, neurotransmitters, hormonal responses, production of metabolites like short-chain fatty acids, or potentially directly through the vagus nerve. Specific for our preterm infants, there are parallel windows of microbiome and brain development. So if this is our preterm birth, say 23 weeks, and this is full-term birth, you can see there is a change in microbiota complexity, also synaptic density, neuronal migration, synaptogenesis. All of these things are occurring over time. And there have been several studies that have linked the microbiome to developmental disorders such as ADHD and autism. And I think one could make the case that perhaps we should think about the microbiome and neurodevelopmental outcomes for our patients as well. So this was another aspect that we looked at for the broader microbiome. We developed the MIND cohort, or the microbiome in neurodevelopmental outcomes. 5 centers: Beth Israel in Boston, North Shore University in Evanston, Illinois, University of Chicago, UCSD, and University of South Florida. Microbiome samples from patients were followed from birth to age 5. And you can get an idea of, kind of our gestational age distribution, average about 28 weeks, the sex distribution, and the race distribution here. And one of the first things we looked at was actually head circumference growth. This is something that's easy to measure and has been associated with neurodevelopmental outcome in other studies. So we looked at head circumference growth over time. So blue is a normal trajectory by Z-score, red is concerning. And what we found was that for patients who were falling off, this occurred right about 30 weeks gestational age. And once they seemed to fall off the trajectory, they continued to do so. There wasn't crossover. So our thought was that if the microbiome was associated with brain development and head circumference growth, that there should be a change in the microbiome about this time as well. So this is just a change point analysis where we looked at the microbiome over time and looked at the number of change points, or how much the microbiome varied in taxa abundance at different times. And we found that the greatest alteration in the microbiome was also occurring right about 30 weeks. And when we tried to model this more specifically, you don't need to read any of these words. What you should see is that we looked at a lot of different factors, and purple is the microbiome. So blue is delivery mode. Orange is morbidities like seizures, retinopathy. Red is antibiotics. Green is nutrition. Way down here is things like birth weight, birth head circumference. You can see the impact that the microbiome does appear to have on head circumference growth. And we could imagine that the microbiome is then also going to have impacts not just on neck, but all of the other conditions of prematurity that have inflammation at their root. Certainly, there have been a lot of studies on the microbiome and BPD. There are studies on the microbiome and retinopathy, and certainly, as I mentioned, there's an association with brain growth as well. So I think as we think about probiotics, we should be thinking more broadly about the microbiome. The microbiome is important in health and disease. And the term probiotics encompasses many different organisms. Encompasses many different organisms. We don't know what these probiotics are doing to the preterm infant microbiome, although this is an area of active research. Under current protocols, the majority of preterm infants that are receiving probiotics were never going to develop NEC. So I think for those infants, we need to think about side effects and other morbidities or long-term outcomes. And we can't say that probiotics are without harm, particularly for those infants who are not going to develop NEC, because altering the microbiome of a preterm infant has impact beyond NEC. Just as a few more points of caution, this is a study on Lactobacillus reuteri, which is another probiotic that has been suggested. You don't need to know what any of this is. You just need to see there's lots of colors. So this is a study of all the different strains of Lactobacillus reuteri and impact on different cytokines. And you can see all the different strains have different effects. Some are good, some are bad, but certainly we need to be considering host origin and strain differences as we think about the probiotics and make our selections. I think we need to recognize that in neonatology, there have been other times that we've been really enthusiastic about therapies, whether that was oxygen or dexamethasone, and found that there were harmful effects later on, just because good things can be harmful at key developmental stages. So in 2021, the Committee on Fetus and Newborn did make a statement. So currently, the statement is that given the lack of FDA-regulated pharmaceutical grade products in the United States, conflicting data on safety and efficacy, and potential for harm in a highly vulnerable population, current evidence does not support the routine universal administration of probiotics to preterm infants, particularly with those with a birth weight less than 1,000 grams. For centers choosing to administer probiotics, the committee suggests discussion of the risks and benefits of therapy with parents, including considering a formalized informed consent process, that there be local guidelines for probiotic surveillance. As I mentioned before, many studies have found that once probiotics are introduced into a nursery, it appears in all of the babies and in the environment, not just those who were intentionally being given the product. And that there is documentation of outcomes, adverse events, and safety, because without data, we're never going to know. The International Scientific Association for Probiotics and Prebiotics does have criteria for a probiotic drug with a specific indication for treatment or prevention of disease. These include having a defined strain of live microbe, proof of delivery of a viable probiotic at efficacious dose at the end of shelf life, a clear risk-benefit assessment to justify use, and appropriate trials to meet regulatory standards for drugs. And we don't have any of that for preterm infants. A study more recently from 2022 did document long-term changes in the microbiome by probiotics. This was interesting. They did metagenomic analysis, so looked at function from 1,431 samples from 123 infants who did not have NEC or sepsis. And they looked at 3 epochs. So earliest, no probiotic. They had another period of time from 2013 to 2016 in which they used Infloran. Infloran has interestingly kind of changed in makeup. So when they used it, it was B. bifidum and Lactobacillus acidophilus. Then from 2016 to 2021, they had yet another probiotic. And then they looked at results. This is the no probiotic, Infloran, and the labinic. And what they found was that probiotics were statistically the most significant factor shaping the microbiome at all time points. So for the Infloran, this green is the Bifidobacterium bifidum. So it's above the others. And this gray is this purple is the Bifidobacterium longum, again, from the probiotic. That's what was given intentionally. But interestingly, Bifidobacterium brev was associated with Infloran, that's this dark blue, and Bifidobacterium animalis was associated, that's this gray, with the labinic despite these species not being named as present in the probiotics. So this again speaks to the lack of regulation and concern with giving probiotics when we're not actually sure. Even if we knew what we were studying, we don't actually know what we're giving. So another point of caution. So overall, my view on probiotics is that we have both an opportunity and a responsibility. Probiotics given to preterm infants do persist and restructure early life the microbiome. Therefore, we do have an opportunity to optimize developmental outcomes. The beauty of the microbiome is that it's modifiable. And so perhaps by modifying the microbiome, we can change outcomes. But I think we have a real responsibility to develop specific microbiome-based therapeutics for specific indications, and I would argue for specific times. All of development is on a time course, and I think our therapy should also be appropriately timed. And we have a responsibility to understand the long-term consequences. So rather than messing with the preterm infant microbiome, I really think we should be protecting the preterm infant microbiome. This includes recognizing that probiotics have effects on the microbiome that we don't understand, specifically with regards to the impact on the broader microbiome community. I think we should be evaluating our therapeutic options in light of an effect on the microbiome and recognizing that there may be effects that we don't completely understand. So this could be things like nutrition supplementation. But overall, the things that we do know, I think are important too, and we should be applying these as much as we can. So antibiotic stewardship and promoting exclusive breastfeeding, I would argue the key thing we can do to optimize the microbiome that don't require probiotics. And I think we could all be doing better there. So I'll end there. I'm happy to take any questions. Thank you.
Thank you, Dr. Claud. That was a wonderful talk. I just wanted to remind everyone just to complete the evaluation via the QR code there. And I also put the REDCap link in the chat. And if you want to chat your questions or you want to raise your hand and unmute to ask your questions, I can help facilitate that.
I'm curious, I don't know if there's a way to take a poll or find out how many of the people online are using probiotics regularly in your nurseries? Do you have a way? I can't really see a broader group.
I think it would have to be a preset poll, but maybe people can just raise hands.
That's fine.
Can people raise their hands if you're using probiotics? We have a trial at our institution. It's not standard.
Okay. And what probiotic are you using? Do you know?
Oh gosh, I'm not sure. Some of my co-fellows are here. I don't know if they know.
Okay. And what is your outcome?
Neck.
Neck. Okay.
Mm-hmm.
And is there a reason that, so do you know what it's not your trial, so I won't put you too much on the spot, but do you know, I mean, because there've been so many trials, what was the thought behind doing another trial? Why is this probiotic thought to be different or better than the ones that have already been tested?
From what I know, and I'm not like completely well versed in this trial that's going on, is it's a multi-center trial and it's focusing just on smaller babies. Oh, one of my co-fellows telling me it's a freeze-dried product of lactobacillus we're using.
Okay.
Yeah. But it's ELBWs, I believe, only.
Okay. And are you guys going to be doing long-term follow-up or just till discharge?
I think I'm not sure about that. I'm not sure.
Okay.
Can I ask a question? Hi, this is Deirdre O'Reilly.
Hi.
Thank you
Good
so
to
much,
meet you.
Dr. Claud, for providing this talk. And I'm sorry I was a little bit late. But I'm curious, when you are asking about trials and outcomes, I agree with you completely. What do you think we can do as neonatologists or people who follow infants to control for the probiotic being the thing that made a difference in terms of the neurodevelopmental outcome? What kinds of things can we look for that will help us in terms of either nutritional status or growth or other things? Or do we have to do stool samples until they go to college?
I don't know that you need to do it for that long a period of time. I mean, honestly, I would love for all of these probiotic trials to just follow up the children to age 5. I mean, I feel like we have standard processes for following up our babies. We understand what this looks like, and there's a broad background against which to test. It's just that it's the reality, right? These trials cost money. And most of the ones I've seen kind of say, well, we don't have the money to follow up these babies long term. I think we just need the data. So that's been my biggest frustration, and kind of this sense that, well, isn't it obvious that it's beneficial? Why would we follow them up long term? They didn't get sepsis. So I don't know that we necessarily need to do anything more than what we would do for many other therapies that we do routinely in the nursery. Just get the data, follow them up long term, and recognize that, you know, the development is important. We're not just concerned about sepsis before they go home. And I do also think we should just do the easies, right? We know about antibiotic stewardship. We could all do better with mom's own milk in all of our nurseries. That's my big plug. It seems so easy to give another drug when we know other things. Even for the first 14 days of life, mother's own milk for the first 14 days of life, I think could make a big difference.
Okay, I think if there's no more questions, maybe Dr. O'Reilly, should we end?
Yeah, I think that's great. You know, if of
Okay.
course, if anybody does have questions that they think of after this, send them either to me or to Dr. Claud directly, but we'll be sure to edit these series and make sure that they're available for everybody online on MedEd on the go. But I really appreciate your talk on this really important topic. Thank you so much.
Thank you. Thanks for having me. All right.
Thank you. Bye.
Bye-bye.